Principal Maharaj of Ramakrishna Mission Vivekananda Centenary College, Honorable Coordinator Maharaj of Department of Microbiology, my fellow colleagues, students, and all the audience connected through live stream. I feel privileged to have the opportunity to introduce our respected speaker of this webinar, which is entitled Gut Microbiome, Brain and Behavior. Dr. Amit Ghosh, J.C.Bose Distinguished Professor and Emeritus Scientist, NICET Kolkata, is going to deliver his speech. Dr. Ghosh studied physics honors in St. Xavier's College, Kolkata and MSc from University of Calcutta. He started his research work on the molecular biology of bacteriophage at Saha Institute of Nuclear Physics, Kolkata. Subsequent to his Ph.D. and after a brief stint as a lecturer in BITS Pilani, he moved first to University of Rochester and then to the University of Wisconsin in USA. He was first to report the presence of actin-like proteins and absence of a major repair pathway in a microbe. Also, his work led to the discovery of many unique features of the genome structure and the life cycle of RNA viruses. Returning India, he, in association with his collaborators at at Institute of Microbial Technology, IMTech Chandigarh, IICB Kolkata and NICET Kolkata, developed an oral recombinant vaccine for cholera using a novel strategy. In 1998, he took over as the director of IMTech, and during his period, it backed CSIR's new idea fund 3 times in a row. The country's first and only international depository authority was established under his directorship. He served as vice president of National Academy of Science, as director of School of Biological Sciences and Biotechnology at Indian Institute of Advanced Research at Gandhinagar, Gujarat. He has published 70 plus high quality scientific papers and guided 14 PhD students. Dr. Ghosh is recipient of various awards and prestigious fellowships and associated with various organizations and scientific research societies. Time is short to mention all this, but enough to motivate our students. I'm sure that today we are going to be enriched with his valuable words, and I request Maharaj to proceed with the session.
Okay, thank you, sir. So today we are very glad to you, sir, to amaze us today, and we are really grateful to you, sir. So we would request Professor Amit Ghosh, J.C.Bose Distinguished Professor, National Institute of Cholera and Enteric Disease, Kolkata, to kindly deliberate his speech on gut microbiome, brain, and behavior. So please, sir, you can proceed.
I would like to begin by saying that I'm really honored, Maharaj, to be invited to this august institution. And I really consider that I'm lucky. And I would like to thank you, Maharaj, Swami Kamalastananda ji, for kindly inviting me. Now, the subject matter of my talk is actually shown in the first slide. And it's a topic which really took off for last 10, 12 years. And I hope that someday some of the students here will decide to work on this area and maybe make some contribution. Now, although this is an emerging area in the sense that it started right earnest not very long ago, but actually the story had its beginning almost 140 years ago with a German scientist called Theodor Escherichia. Now, you know Escherichia through Escherichia coli, and even in the heyday of when Koch was propounding his theories, Escherichia decided to look for the microbes present in the human stool because he believed that through this analysis and through this characterization, maybe you'll be able to solve some of the intestinal disease problems. Now, you know, since the days of Escherichia, much time has been spent, and particularly after the advent of the deep sequencing methods, now we know that human body is home to trillions of microbes. This is a cover page from a Nature, and you can see here it's a rough presentation of what are the different microbes are present in the different places of our body. Now, I just want to bring the attention of the students to two terms, microbiome and microbiota, even though they are often used in interchangeable manner, but they have very distinct meanings. Microbiota is the microorganisms that live in an established environment. For example, if I talk about the microbes that is residing in my gut, I talk about gut microbiota. And microbiome is a full complement of microbes, their genes, the genomics present in a particular environment. Even though intense research is going on in the microbiota and the different parts of the organism, the most attention or the most work that goes on is in the intestinal region. It's a rough presentation of how these are distributed, and also on the left side of the slide you can see what are the different microbes which are supposed to be present, which has arrived at by deep sequencing. Now, in spite of all of this, it's now known that gut microbiome is largely defined by two bacterial phylotypes: bacteroids, mostly gram-negative, and firmicutes. Together, they make up for more than 99% of the gut microbiota. But then there are others also: proteobacteria, actinobacteria, fusobacteria, and others. Now, since I think many of you are microbiology students, you already know that gut microbes perform a variety of functions. This cartoon just shows roughly, in a very simplistic way, what are the different functions they perform. We all know that human body cannot make many vitamins, which are made by our gut microbiota. They stimulate the immune system. They also fend off the pathogenic microbes. So all these functions are basically done by the gut microbiota. So this is what I was talking about. If you look at the number of publications that has come up, this is from up to 2016, but the graph goes up. In 2002, you could see there are very few publications on the microbiome research. But by 2016, we had about 3,500. And today, if you go straight, it is close to 8,000, if I'm not mistaken. It's just like that. Now, what I want to do is, in developing the story that I'm going to tell you, I intend to follow a historical path to highlight two aspects of scientific progress. I think this is important because today, with this multiple choice questions and other things, often we lose sight of this thing. For example, when you look at Watson's book and read about transcription, it looks so simple, but it's not so. Tremendous amount of work, failed experiments, many people's efforts have gone into where we stand today. You know, the progress of science, actually science progresses through trial and error. And as I just now said, behind each scientific truth lies the efforts of many. Often failed experiments generate clues which help one to design right experiment. And second point that is also to be remembered is the progress of science does not follow a linear path. One does not necessarily jump from one conclusion to the other. Often a new path emerges when divergent observations coalesce together. And in this case, four divergent observations carried out at different times by different people have come to take us to where we are today. First is mind has influence over gut function. I'll elaborate on all of this one after the other. Secondly, the gut has influence over mind. Third observation was some pathogenic microbes can affect mind. See, these are kind of unconnected. When I talk about the third one, the some pathogenic microbes, but we'll see how these led to some very important observations and very important conclusions, which has helped us to arrive at where we are today. And then after the advent of deep sequencing, we now know that gut harbors trillions of microbes. Now, our story actually begins almost 140 years ago with this man called Alexis St. Martin. Alexis St. Martin was a 19-year-old Canadian who was a fur trapper. And at that time, he was working in a small place called Mackinac Island in the United States. And in course of the work, one day, he got shot accidentally by a bullet. Now, this is a small island. There are no doctors, but there was an army camp. So St. Martin was taken to the only doctor, the surgeon who was present there. Sorry, his name was William Beaumont. So Beaumont, what he did, he performed a number of surgeries on this person. The first one to save his life, and later on to repair the wound. But what happened that after he performed quite a few surgeries, this fellow, St. Martin, refused to undergo any more surgery. He said, enough is enough, and just leave me alone. So what happened was that a gap, a fistula, remained on his body. Now, this is at that time, there was no photography. So this was a picture that Beaumont did. And this hole or the fistula never got infected because, as you know, stomach is acidic. And the acid, stomach acid, constantly irrigated this wound. And therefore, it was not infected. But this man, William Beaumont, was a far-thinking man. He immediately realized that he has been presented with an unique opportunity, so he could really observe what goes on inside the human stomach. And at that time, very little was known about our digestive process. People thought we have grinders in our stomach, just like we chew food. So there are grinders in the stomach. When the food goes in, they're chewed up by these grinders. So what this man did, he would hang a small piece of food through this hole into St. Martin's stomach and watch what happens. And he realized for the first time that there is no grinders and digestion takes place through some unknown chemical means. And then he documented this, but one observation that he did, this is the book he wrote, but one observation he made was very, very important. He showed his experiment 34 described in the book. He says, the circumstance of two may also have contributed to interrupt the progress of digestion, such as anger and impatience, which were manifested by the subject during the experiment. That means if St. Martin was angry for some reason, this digestion process got hampered. And similarly, he was impatient, same thing happened. So this was the first clue that came that maybe, the emotion, otherwise in the brain, has some control over our digestion process or the gut functioning. And then this was taken forward. This was a very hot field. And this man called Walter Cannon, considered to be one of the founding fathers of gastroenterology, he wrote a very influential book, The Influence of Emotional States on the Functions of the Alimentary Canal, in the American Journal of Medicine. So what the take-home lesson from this was that mind has influence over gut function. So this was an independent observation and it stayed there. But the question is, what's the reverse? Converse is also true. Does gut influence mind? Now, not infrequently, doctors observed that chronic bowel disorders cause anxiety and depression. We all know it. If you have a persistent diarrhea, oftentimes you don't feel good. You are in sour mood and things like that. But this was an anecdotal kind of an experience. There was no solid experimental proof the way Professor Beaumont, Dr. Beaumont did. But take-home lesson from here was that gut function has influence over mind. But large number of clinicians made this observation. So this was something that could not be easily discounted. And then there was a famous philosopher, uh, William James. He also wrote a paper on this, but it was mostly speculative. And then this doctor, French doctor, Charles Bouchard, he wrote a book and where he claimed that mental health disorders could be due to chemical breakdown products of bacteria action on food material. Now, this was discarded subsequently, but as we shall see, that maybe there is some truth in it. Now, the question is, these observations were disputed, as I've said, but they could not be discarded altogether. The third line of clue came from the observation that there are pathogens, pathogens who can affect our behavior, who can affect our mental function. I just give two examples. First is Lisa virus, rabies, jolatonko, hydrophobia. If one gets rabies, oftentimes the victim of the biting becomes violent. He starts barking like a dog. In other words, this Lisa virus, as it moves through our nervous system, it profoundly affects our mind and affects our brain. And I am assuming that brain is the seat of the mind, even though there are some, other observations on that also. And second is a bacterium which causes a disease called syphilis. The people who suffer from this disease get often demented. So there are two proofs here that there are two pathogens which can affect our brain and our mind, and there are other pathogens as well, but these have violent manifestations. So the question is, subsequently it was discovered that there are other pathogens which do not manifest in such violent forms. They give mild infection, but they can affect our mind. And a good example of this is this parasite, Toxoplasma gondii. This is an intercellular parasite. It is a crescent-shaped organism. It can affect all mammals, including man, everybody, human. But it only grows inside the intestine of the felines, cats, and others. Now, what happens if a human being gets infected by Toxoplasma? Then I'll come to this slide a little later. Let me say first what happens. We get very mild fever, it goes away. Mild in this position for a day, and then it disappears. But what happens is, it does affect our behavior. I'll talk about the cat a little later, but there are some observations which scientists noticed that, first of all, the schizophrenic patients have 2 to 3 times more antibodies against Toxoplasma gondii. So if one looks at the antibody profile of a person who's schizophrenic and compares it with those of a person who's normal, it is found that Toxoplasma gondii antibodies are 2 to 3 times more. Second observation was that the drugs which are commonly used for the treatment of schizophrenia, are also effective against toxoplasmosis. Third was a very interesting observation which was made in Czechoslovakia by the Czechoslovakian army. They found that some army recruits get involved in car accidents much more often than some others. And what they found was that these army recruits, which fall into car accidents 2 to 3 times more than the normal recruits, have been infected with Toxoplasma gondii at some time. And even though afterwards there's no trace of this parasite was there, but the effect remained. Now, when this was presented as an example of some microbes, some organisms affecting our mind, the critics said, well and good, the first examples were highly pathogenic organisms, and now you're talking about a parasite. Those organisms, those microbes, Lissa virus or the syphilis causing microbe, they're not gut bacteria. And secondly, Toxoplasma gondii is a parasite and is not present in the gut. So the next crucial experiment comes from the work of a man called Charles Light. He shows that, I just cannot resist this showing this. You see, as I said, that Toxoplasma gondii grows in feline gut and it can affect all the mammals. Now, this is a normal cat. What is a cat's behavior? Cat will, eat up this mouse. So the mouse avoids the cat. But when a mouse gets infected with Toxoplasma gondii, fear that the mouse has for the cat completely disappears and it's permanent. So even after the mouse gets cured, is still not afraid of the cat. So here is a very, very good example that there is some microorganism which can cause a permanent change in the behavior pattern. But as I said, the critics said that all these are fine, but Toxoplasma gondii is a parasite. It's not a microbe. And the other examples, they are also not gut microbes. So the next crucial experiment came from this gentleman, Light. He is a professor, I think, in the University of Texas Austin campus. He showed that if a mouse gets infected with a subclinical infection, so the major diarrheal manifestation is not there with Campylobacter jejuni, then what happens? It changes a behavior pattern. So here is a microorganism which also happens to be a human pathogen. It's a diarrheal gut pathogen, and it can cause a change in behavior. It's a gut pathogen. It's a microbe. So the next forward step was that a microbe, which is a gut pathogen, can cause a change in the behavior pattern of an organism. So the observation so far is pathogens, including the fairly innocuous ones, including gut pathogens, can induce behavioral changes. That is, gut has influence over mind. But the problem is, now, but what about the microbiota? They are resident microbes which are in our gut and they are not pathogenic. So do they have any control over our behavior and pattern? That is the subject matter, is the talk. Now, as you know, there are about 200 to 600 million neurons in the human gut. And there are 10 to the power 14, that is 100 trillion commensal microbes also in our gut. So some scientists argued that if they're in so close proximity, there has to be some crosstalk. It was a hypothesis. And they thought that, judging by the previous experiments that I have described, it is unlikely that these microbes, trillions of them sitting inside our gut, do not interact with the neurons which connect to our brain. But it was still at a hypothesis stage till a Japanese scientist, Nobuyuki Sudo, performed a milestone experiment. What he did was, this is his paper. So I'll tell you what he did and why this paper is considered to be a pioneering paper in the field. But before I describe Sudo's work, I have to tell you about some animal models Sudo used. And these are shown in the next slide. The specialized mice are used in such studies because the idea is we want to see what gut microbiota that are present in the human gut, how they influence the brain and the mind. So scientists have created several classes of such mice. First is germ-free mice. These are also called axenic. They're free of all microorganisms. Second is specific pathogen-free mice. So here, a particular pathogenic microbe is absent in the gut of such mice. And then there are notobiotic mice. These are axenic mice or germ-free mice in which specific microbial strains have been introduced depending upon the need of the scientist. So what Pseudo did was he compared the germ-free animals or mice with animals colonized with specific pathogen flora. And then he also perturbed the stable microbiota in the healthy mice by oral administrations of antibiotics which are not absorbed, because it is absorbed and it goes to the bloodstream and other problems will come. So these are non-absorbable antibiotics like a neomycin bacitracin combination he used. And what he found was, I'll explain in the next slide, but before that, let me just say that some very key players which are thought to be responsible for this gut-brain communication. One is vagus nerve. It's a long nerve that links heart with GI tract and lungs to the brain. It has several branches. Then hypothalamus is a brain region that controls emotion. It controls functions like hunger and thirst. And then scientists imagine a hypothalamic pituitary adrenaline axis. I'll talk about a little more later. That includes all of this, and it controls the responses of our body to the stress. And finally, cortisol is a hormone which is responsible when we get stressed. This hormone gets generated. So what happens is shown here in the cartoon form. See, first of all, whenever we are stressed, the hypothalamus makes this hormone CRH, and then it signals the pituitary gland to make this adrenocortical, it's a long name, TH hormone. And then that goes to the adrenal glands, which are in our kidney. And then kidney produces the cortisol, which takes care of the stress system. See, what happens is more sugar metabolism takes place. You see, when people are stressed, they tend to eat more sugar, I mean sweet. It helps. So these are the kind of thing what happens when we are stressed. Now, what Pseudo found, what he did was he just measured these hormones, And what he found, I don't know if you can see this very correctly. You see this hormone ACTH, if it gives in a stress, it increases and then it comes down. So this is the same for another hormone. But what he found was that depending upon the stress, these hormones get generated. Lower graph is a control, upper one is what happens in the experimental animal. He also found that it's stress specific, that not all stress produce this effect. But whatever it was, it is considered to be a milestone experiment because it showed for the first time that there is a direct connection between gut and our behavior or the mind pattern. The stress response was more, this is what he found, in adult germ-free mice compared to control mice with normal composition. So if a normal mouse is there with a normal gut microbiota, it feels less stress compared to a mouse which does not have any microbe resident in its gut. So the conclusion here is then gut microbiota must be helping in some way to cope with the stress. And manifestation of that was what you saw in the previous slide, generation of stress hormones. So microbial content of GI tract influences the development of stress response. That was the main observation. Now this simply shows the summary from what Pseudo's paper showed, that compared to young, okay, you may not put too much attention on this. The only thing I will draw your attention to the third one from the top in green, we showed that the effect of stress could be completely reversed if this germ-free mice was colonized with Bacillus infantis. So there is a particular microbe which is considered to be, today you hear a lot about probiotics. If it is given to a germ-free mouse, the stress the germ-free mouse felt can be completely reversed. Now, basic observation, if you forget about all the details, are two. The microbial content of GI tract influences the development of appropriate stress response. And then there is a critical window. That is, if it is done with adult mice, this doesn't happen. So it has to happen early in life. Now, this has been considered to be the number one experiment in the field, which actually directly scientifically showed a relation between the gut microbiota, mind, and hence the behavior. But what it didn't show, it did not show, it provided indirect evidence, because what Pseudo did was he measured the stress response by measuring the changes in the level of stress outpost. But he did not address the phenotypic issues. His experiment did not tell how the mouse behaves after the beta infantis is, bacillus infantis is given back to the unfree mice, how it behaves. So this gap, that is from measurement, stress measurement through the measuring hormones to behavior, was, it took 7 years, and three independent groups working in Ireland, Canada, and Sweden, they performed crucial experiments. But before I explain to you the experiments that they did, I have to spend a few minutes telling you what's the experimental setup. Now, what you see in front of you is called an elevated plus maze system. It is nothing, it is two kind of, as you can see, cross parts. It is elevated so that mouse cannot run away from the floor. One, this where the mouse is sitting, this is open. It is open to the environment. But the one which is at right angles, it is walled up. So this is basically a combination of an open space versus a closed space. Now, normal mouse is very curious animal. You know, it loves to explore the environment. It'll go everywhere, but eventually, it likes to be in a dark place. So it's a behavior pattern. If a normal mouse is there, what will happen? It will first explore, as this particular person is exploring. He is in the open space. And then afterwards, he will or she will return back to those closed areas. And this is a variation of the theme. It's a light and dark box. It's an open area, which is lighted area. And there's a dark area. Again, if a mouse is left here, first of all, he or she will explore all the surroundings and then eventually try to go back to the dark area. Now, as I said, the normal explores both light and dark areas and stayed in the dark, mostly in the dark areas. But when they used a germ-free mouse, they found that they spend more time in the lighted area. Now, lighted area is considered to be stressful, because everybody wants to avoid situations which they don't like. But here, what is found that this germ-free mouse is spending more time in the lighted area as opposed to the normal mouse. Now, so here again, a direct proof that if you take if you take a mouse which does not have gut microbiota, its behavior pattern changes. Now, here you must see that there is a contradiction between what these people found and what Pseudo found. What Pseudo found was that germ-free mouse is more afraid of stress. Here it's the reverse. Here the germ-free mouse spends more time in the lighted area, so he's not afraid. Now, initially it created a little bit of problem because both experiments were done by very established scientists and they did very careful studies, but later on it was attributed to the gender difference of the mouse. So depending upon the male mouse or female mouse, this change in behavior pattern is observed. But the next key experiments were done very convincingly by the same group of scientists. What they did was they took two classes of mice. One is bulb C mice. I'm sorry, there should be one B there. It's not Balsi, it's bulb C mice. It was created by a man called Hangley, Bagel Hangley or something, that now comes. And these mouse are immunocompromised. They are very timid. And if you hold them and put them in one place, they stay there. Whereas, and these are inbred mouse, so the litters are made to breed with each other. As opposed to them, there is NIH Swiss mice. They are outbred. I mean, they're normal mice and they're highly exploratory. Again, they are not allowed to mate in the wilderness, but within the setup, they are what they call as outbred mice, and they are highly exploratory. Now, what was found that if a germ-free bulb-C mice is colonized with the microbiota from NIH Swiss mice, then they behave like NIH Swiss mice. Let me repeat. A bulb-C mouse is there. It is timid, less exploratory. And now scientists take the microbiota from the Swiss mouse, which is highly exploratory, puts it into the intestine of the bulb-C mice, and lo and behold, this bulb-C mice becomes highly exploratory. That means the microbiota induces a massive change in the behavior pattern. It's just one more confirmation of the early experiment with elevated maize and other things. So here again, the conclusion is host behavioral phenotype is also influenced by microbial factors. Now, another group of scientists, I think his name is Berry, he took it one step forward. He made it examine, these are, the overall behavior pattern, whether the mouse becomes more exploratory or less exploratory, mouse becomes, wants to go to the lighted area or the dark area. But this gentleman wanted to see if it changes its social behavior. And what he found was that germ-free mice are significantly socially impaired. They are kind of, there are human beings also don't like to mix with people. They like to be with themselves. So germ-free mouse are also like that. They're socially impaired compared to the pathogen-specific mice. Now, what is the experimental design? Let me show it to you. Now, these are three compartments. Let us first look at the top panel. So, middle chamber, one puts the test mouse. That is, it can be a germ-free mouse or it can be a wild mouse. And one side, there's an object is placed, an inanimate object, and another chamber, that is the first chamber from the left, another mouse is put. Now, what happens is, if a germ-free mouse is placed in the middle chamber, then it just is not doesn't like to spend time more with another mouse. It rather spend time with the object, inanimate object. So let me repeat what I said, that a germ-free mouse is put in the middle chamber. It just spends more time with that inanimate object rather than with the other mouse. Whereas a conventional mouse will be less interested in that inanimate object and will try to spend more time in the chamber, which has another mouse. Next experiment was again a little more finesses added. Here, what was done was, in the middle chamber, the test mouse was put, but on the first chamber, a mouse was put which is familiar to the first mouse, that they are from the same cage, spend some time together. And on the right-hand chamber, they put a novel mouse. So what was found was that the conventional mouse likes to spend more time with the familiar mouse, just like we want to spend more time with a friend rather than somebody we don't know. So the conventional mouse wants to spend more time with the familiar mouse and doesn't pay much attention to the novel one. Whereas a germ-free mouse doesn't care. I mean, he is completely neutral. Which way he goes, these are all, very carefully videographed. It's found out that they just don't care. They can be here, they can be there. So again, there is one more proof that gut microbiota definitely helps our behavior pattern, not just in the stress or trying to find out which way to go and which way to avoid stress, but also in our social, not in our, but the mouse's social behavior. Next came a very interesting observation. Okay, so I've already said this. they found that if they put this lactobacillus strain, again from a probiotic, then this mouse changes its behavior pattern. So the gut microbiota, germ-free mouse, if it's fed with this strain, then it starts behaving like a conventional mouse. And this table, you need not bother. I'm just listed behavioral phenotypes of different microbial composition. So after these basic experiments are done, scientists all over the world make different cocktails because they know what are the microbes present in the gut through this deep sequencing. And they did all the experiments and tried to find out how their behavior pattern changes. We are not mouse, we are human beings, and the question comes that this is all fine, and all these experiments have been done with mouse and animals. What about human beings? So a very interesting observation has been found. I just give one example. What is the time? Okay, I've still not 10 minutes. See, autism is something you may be familiar with. Autistic children, they're kind of emotionally restrained, is a developmental neurobehavioral disorder. They don't want to interact socially with anybody. They don't like to communicate to anybody. They're highly intelligent, but this is a pattern. And there appears to be a link between the gut microbiota and autism. The first idea for this came when people found that the autistic children invariably suffer from a lot of gut intestinal problem. So it gave some thought to some scientists that is it possible that autism has some link with the gut flora? I told you in the beginning, I showed you a slide, but here I just want to re-emphasize the point that it's largely defined by two bacterial phylotypes, bacteroids and firmicutes, and then there are others, proteobacteria and others. So I'm saying this because what has been found in case of autistic children, this was a very work was also controversial, but nobody could actually show anything wrong with it. They found that the autistic children have higher proportion of bacteroids, and they have lower levels of bifidobacteria, higher levels of lactobacillus, and other similar levels are there. So this was controversial because people thought that if that is the case, then feeding children with the lactobacillus and probiotic, it can be cured. And some companies, there's all they do, they started advertising about it. But anyway, nobody could actually question the data that was obtained. And then with adults, this experiment was done in very recently, about 2 years ago. This was done with Belgian, 1054 Belgian and 1054 Dutchmen. And out of these 130, 73 people were diagnosed with depression. And when they were analyzed very carefully, it was found that two kinds of bacteria, Cropococcus and Dialister, were found to be missing in depressed subjects. So when the gut microbiota of these people were suffering from depression was analyzed with deep sequencing methods, it was found that compared to the control people, the normal people, these two bacteria were absent. And the same observation was made with 1054 Dutchmen to show that this was nothing to do with country specificity or ethnicity or whatever. So this is another one proof that our mental well-being is intimately linked or behavioral pattern with the gut microbiota. Now this you have seen in a different form before, that all right, all these are fine. So we have a phenomenological observation, phenotypic observation, which proves beyond doubt that there is a communication between the gut and the brain. And brain and gut communication was known for a very long time. It was known through the days of Cannon and Beaumont and others. But now it is very firmly established that there is a channel, a pathway from the gut to brain, through which gut microbiota perhaps communicates. And these are the main players. Are the vagus nerve is there. I've already mentioned about it. And then they talk about the hypothalamic pituitary adrenal axis, which I showed in connection with that stress slide. What happens? Now, this is a very simplified picture of the key players which are involved in gut to brain communication. You look at the box panel, it shows the nerve is touching the nerve cells, and then the immune cells are there, the blood vessels are there, and gut microbes is inside the intestine. So what happens is when, as I said, there are crosstalk between the gut microbes and the neurons. So how this pathway is used up in sending the signals is shown in the next slide. Now, I must caution you here that some of it is speculative. That is, in the sense that there are very telltale evidence, but solid scientific evidence in some cases is not present. So there are gaps, but scientists are pretty sure that pretty soon those gaps will be completely covered. So the three major pathways are envisaged, and first two pathways are already proven. The third one is a little bit of, still it has to be shown. The first one is, they first produce the neurotransmitter. See, what happens is our gut cells can produce serotonins. 90% of all the serotonin, that the growth hormone, which causes our emotional, has a big control of our emotional state, uh, is made in the gut. So what happens is gut microbiota sends some signals to the cells in the gut, which lines the epithelium, and then these cells produce serotonin. And this serotonin activates the vagus nerve, and the signal goes to the brain, to the hypothalamus, and we feel happy. Or you may have heard about a medicine called Prozac, which is heavily used for emotional control and others. The second one, the largest number of our immune cells are present in the gut, and they produce cytokines. And these cytokines, again, travel through the blood. They get dispersed in the blood, and then blood goes and all over our body goes to crosses the blood-brain barrier and goes there. But again, here, as I said, this is not strictly proven. There is telltale evidence, but solid scientific evidence which will be accepted by everybody is not there. And then comes the third one. When we eat something like some starchy food, or we generate short chain fatty acids, and these short chain fatty acids then stimulates the gut epithelial cells, which then again produce neurotransmitters, and through vagus nerve they go. This part also, of course, is fairly well established. Now, I'm showing the same thing for your convenience, that microbes in the gut produce neurotransmitters and metabolites like butyrate to circulate to the brain, where some kind of then small enough to cross blood-brain barrier, other all cell activity. Then second is microbes interact with the immune cells of the gut, and prompting the cells to make cytokines that circulate from the blood to the brain. And then I have mentioned that they have produced neuroactive molecules and peptides, and same through the vagus nerve activation. They go and make our feel good or bad depending upon our emotional state. Now, what are the future directions of research? Now, here, in a broad canvas, these are all well established. What are not there are the gaps which needs to be filled up, and scientists are quite confident that they all can be filled up. But there are areas, like, for example, I told you about Pseudo's experiment contradicted with the experiment done by the letter workers, and then contradiction was explained by saying that it is because of the gender. Now, these are the areas where very active research people are trying to do, and these are the future directions of research. First is influence of sex difference, because male and female, they have fundamental differences, and whether they can affect, microbiota brain communication. Do they have any effect on learning and memory? You remember that the microbe which causes syphilis causes dementia, but that is not a gut microbial, and that is a pathogen. So it's just like they found that there are a particular absence of particular gut microbial causes depression, whether there are some strains in the trillion that is inhabiting our gut has some effect on learning and memory. I have not said anything about the gut microbiota and CNS development because that's a separate seminar topic. In fact, for our brain development, it is plays a role. Initially, people thought that the, the, the microbiota is acquired by humans during birth as a pass to the birth canal. They get from the mother all the microbes. They eventually inhabits the gut, but now it's not known. It's not known. So even the inside the mother's womb, there are microbes. Secondly, uh, what role do gut-brain axis play in childhood neurodevelopment disorder? So this is a very, very important area because if people can really find what microbes are absent or what microbes are present in excess, that leads to or can lead to autism, then that will be point towards a treatment from a very, very dreaded kind of a situation because the parents of autistic children suffer tremendously. But the children suffer definitely, but the caregivers, the parents, for lifelong, it's a tremendous problem. And there's no treatment. So therefore, if these are someday discovered, that will be a boon to humanity. And if you look at, I'm very fond of telling my students that, when today you can, like I'm talking to you, my computer is not connected to anything, it's a Wi-Fi. And go to astrologers, we go to this place, that place, but we are not simply get excited about what science has given to us. It's a tremendous kind of a thing, and we don't just feel excited. At least I found many students don't get excited. They will be using their smartphone all the time. It doesn't excite us. What excites them are all kinds of X, Y, and Z things. I'm not saying that it's not good to be excited about when Virat Kohli does a century, but I think that we should be equally excited that when I was a student, these were unthinkable. But today it's possible. So I'll just conclude my talk with a quote from Richard Feynman. Many of you may have heard about him. He is one of the greatest scientists last century produced, and he, of course, died in this century. He said that progress in science is often built on wrong theories that are later corrected. So people should not be afraid of failed experiments. And unfortunately, when the project is written, we have to paint a very rosy picture. Otherwise, the funds do not come in. But actually, science doesn't progress that way. Science progresses where the failed experiments are equally important as the successful ones. And I thank you very much for patient listening. I thought I've been able to keep my time. Thank you.
Okay, thank you very much, sir, for your wonderful and encouraging presentation. Now I request our students to please present your questions, your queries to sir. So please deliver your question.
Am I audible?
Yes, you
Yes,
are.
Sir, my question is what is deep sequencing? I couldn't
Oh,
understand.
okay. Sorry. See, what happens is initially, if you wanted to know what's present in a culture, let's say in stool, what you would do, you'd plate it. And then you will see some colonies and you will decide that these are the ones which are present. But then what happens is, there's a thing called great plate anomaly. Not everything grows on every medium. In the laboratory, you use a particular medium. So maybe the bacteria, some bacteria likes to grow in there, but there are numerous others which will not grow. So you will never know about their presence. Now, after the sequencing methods came, people realized that they do not have to simply culture. What they can do is they can take whatever DNA or everything is present in the gut or anywhere and do a sequence. And from that sequence, they can come to the conclusion that what are the different kind of microbes that are present. So deep sequencing simply means a method of sequencing a, see, what is the problem is you got a mixture. You got a mixture of gut microbes and other DNA. Some you can culture, some you cannot culture. You lyse them, you get DNA and you sequence them. Now how do you know how many different bacteria are present there? So for there are some methods by which there are some telltale signs. One is the ribosomal RNA sequence, which tells you whether different kinds of microbes are present or not. This is in very simplistic form the explanation of deep sequencing. Am I clear?
Yes, sir. Yes, sir. Thank you, sir.
If you have not, I will try to explain once again.
What I have inferred from it may be a type of trial and error method, that what are the different types of microbes that may be present. I thought like that. Am I right, sir? Or
No, you see,
by
you asked me
looking at
about
the
deep sequencing. So again, let me
uh,
try to explain it in another way. So what do you do in a lab? If you're a microbiology student, you plate it and you see the bacteria
hmm,
and you count them and say, okay, it's E. coli, it's Vibrio, this and that. But there are
hmm,
microbes which you cannot culture because, like,
yes,
I cannot survive on, let's say, Eskimo diet or the diet which is, I mean, what a cow eats, I cannot eat. So if you give me
hmm,
cow feed, I'll die. A cow will not be able to eat my food. So similarly, all microbes cannot be comfortably grow on all the media that you give. But sequencing allows you to sequence the DNA. And by sequencing the DNA, you can come to the conclusion that what are the unknown sequences present there. Then you look for the ribosomal RNA sequence because that's a telltale signature
hmm,
for a particular kind of a bug. And then you come to the conclusion that there are so many different kinds of microbes, those are present. And this method of doing it's called deep sequencing method.
yes, sir. It's clear.
Hello, sir.
Yes, please.
My name is Samrito Acharya.
Haji Maharaj.
Firstly, I would like to say that it's a great honor that I'm getting this opportunity to interact with you. Sir, my question is that, as you have told, that there is a link between psychological behavior and the microbiota present in our different tracts, different parts of our body. So my curiosity is that as we humans evolved since prehistoric times, so is there any linkage between this microbe to the evolution for human species, and does it does there is any linkage between our behavioral changes since prehistoric times?
Um, Actually, there's a very interesting paper that has come out in Nature. They have really tried to analyze that whether, along with our evolution, whether this microbiota profile has also changed. Because you see, the human beings have evolved in a particular, different, there was a Neanderthal man, there's this man, Cro-Magnon, this and that, and whether the microbiota evolution also changed. But it's a very difficult research area because one can from the dead tissues isolate DNA and then do the sequencing, but to get a complete picture is a very tough proposition. So this paper, if you're interested, I don't know, we call Swamiji's Maharaj, I don't know how the brothers are to be called. So if you're interested, I can send you this reference and you may like to read that paper, so you'll get some idea from there. So if you are interested, then I can send it to Swamiji and this reference, and then you can take it from him and read it.
Yes, sir. I will be glad if you get
Yeah,
that.
I'll do that. I'll just note down your name and I'll send it to. Or just give me a minute. Yes, Brahmachari Maharaj. I will do that. Surely I'll do that.
Thank you, sir.
You're welcome.
Hello, sir. Am I
Yes.
audible?
Yes, you are.
Sir, I'm Kaushik Bhattacharya from 5th semester microbiology. Sir, I actually have two questions.
Yes.
First of
You
all,
are a student there or a faculty?
no, sir, I'm a student, sir.
Oh, you're a student. Okay. Which year are you in?
Sir, I'm in 5th semester.
Fifth
I
semester.
mean,
Okay.
last
Yeah,
year.
please go ahead. Please go ahead. Yeah.
Sir, I actually have two questions. First one is
Yes.
from your presentation, I have generated an idea that Toxoplasma gondii is shown to be as a zoonotic disease.
Mhm.
So what is the mode of transmission of this disease? I mean, how
It's
it
just
comes from?
if you have a feline, if you harbor if a cat pet and you handle it, and so from the cat from that pet's excreta you touch it and it goes through your mouth, and that's how it happens.
Okay, sir. Okay, sir. And another question is, sir, does cortisol play any role in developing diabetes in human? I mean, as you said that cortisol level is getting high.
I cannot answer this question right away. Uh, I think it does, but I would not venture to give you a correct answer because I have to double check it. Because stress is connected, with diabetes. If you are very stressful, then, this old age
Yes, sir.
diabetes is often because of that. But whether cortisol plays a direct role in that, I would not like to give you an answer which is half baked and not thoroughly checked. So I can double check it, and if I find an answer, I will certainly be able to communicate. But right now, I will not try to venture. But my guess is yes, because stress is one of the causative agents for diabetes. If you're very stressful, we all know the sugar level goes up and all kinds of things happen.
Yes, sir. Yes, sir. All right, sir. Thank you.
You're welcome.
Hello, sir. Am I audible?
Yes, you are.
Sir, I am first year zoology student, Rajashri Chattopadhyay. So
Okay.
my question is, can you please highlight a summary of the relation of gut microbiota with CNS?
See, as I said, that you can see from the central nervous system, vagus nerve plays a central role. So some of this I've already mentioned, but in greater depth, this is an emerging area of work. People are trying to find the nuances of it. But as far as the central nervous system is concerned, the vagus nerve plays a central role. And whatever you see, whatever I have said, and whatever the experimental evidence is there, you have seen that gut microbiota, whatever reaction, other than the one cytokines which goes to the blood and all that, the main conduit is vagus nerve. So that is the main conduit through which it influences the behavior pattern.
Thank you, sir. And
Well,
sir, one more request, sir. If you can please send the report to me too, that you are going to send to Samritho, I would be very glad.
what I can do, if Shamiji wants, I can send you the entire, the slides that I have presented today. That will be of any help. And the only thing I'll do is I'll insert the references, because I didn't mention the references. I can do that, and but that will for that you have to give me some time, because the slides I can dispatch very easily, but you have to put in the references. Then I have to actually put in the references, send it to you. So what I can do is I send it to Swamiji, and along with that reference for that paper, which is Parachaitanya, Brother Parachaitanya wants, and this you can take from the Maharaj, and you can look at the slides yourself and decide whether it's going to be of any use to you or not. I can do that. I have
It
no
would be
problem.
very helpful for us.
I will do.
Definitely, it would be very
I
helpful.
will do that, but you have to give me a week's time, because these kind of. I jokingly said after this virtual mode has come, we get very little time, because previously, if one arranged a meeting or a talk, one had to go there. But today one doesn't have to do. So you almost every day there is one request for a talk or some meeting or something. So you have to give me about a week to 10 days time. And I promise that I'll send all of you to Maharaj, and from Maharaj, all of you can take that. You can take the slides and you can take that other I mentioned about the evolutionary context of the microbiota evolution. I will do that.
Yes, sir. Thank you in
I am
advance.
Santanu. Hello, sir.
Hello. Yes, please.
Hello, sir. Can you
Yes.
hear me?
Yeah, I can hear
Sir,
you.
sir, my question is that any diseases which are related to the brain and the nervous system can affect our gut microbiota.
So you are asking
Is
that
there
if
any
the,
relation? Means, sir, is there any relation?
no, so what you are asking is whether if I have a brain disease, whether it affects our gut microbiota. Like it will all depend on the kind of disease one is talking about. If it is a disease like, say, meningococcus, the answer is yes. But it is something, if it is, let's say, brain stroke, then its effect will not be direct, it will be an indirect effect. So it all depends on what kind of disease we are talking about. See, if you have a disease which affects the central nervous system, affects your, let's say, hypothalamus, we also saw that much of it, the traffic is two ways. So one path, if you block, obviously it will have some effect on your gut microbiota. So it all depends on the kind of disease one is talking about.
Thank you, sir.
Thank you.
I am Santanu Khuila.
Yes, Shantanu.
How my daily diet affects gut microbiota?
This is one area where Indians have done a lot of research. See, ultimately, your gut microbiota will be depend on your lifestyle, your diet, and so many other things. For example, the people in Nagaland, they eat insects. Okay, that diet, and they also eat all kinds of flesh products. That is very different from what let's say South Indian Tamil Brahman eats. So their gut microbiota profile is different. But what happened was some studies was done with some African children. Gut microbiota was studied and compared with those of the Italian children, and considerable difference was found. But what was found was that there is something called a core microbiota, and there's one scientist, he divided or differentiated the entire human types as 4 enterotypes. That is, depending upon what your diet is, where you are. And then for some time it was thought to be a very, very, what to say, crucial kind of evidence. But later on it showed that it is not really so, because in a country like India, where people have so many different dietary habits, the gut microbiota profile, there are 4 places in India where their active research is going on. One is, Translational Research Institute in Faridabad. One is a laboratory in Guwahati, that's a D.Sc institute. One is in down south in Tiruvananthapuram, and the 4th one is in Pune, National Institute of Cell Science. So what they do is they just look at the people who have the different diets and different lifestyle, and try to analyze their gut microbiota, and they find that is very, very different. They cannot be grouped like the way you can simply group, let's say, a Bengali with vegetarian Bengali versus a non-vegetarian Bengali. That kind of thing you cannot do, because ultimately these have to be dependent on the human physiology. If you are eating something very different, we are leading a lifestyle which is very different, it has to affect the microbes that reside in our gut. So this is where Indians have done a lot of work, because other works with the kind of work which I mentioned are difficult to do. They're trying to do it, but the whole setup and the approach is difficult. But this work on gross level, that how the gut microbiota correlates with our food habits, very active area of research in in the country.
Which type of diet is most important? I mean to say that which type of diet is
See
works best?
see, there's an American study. They say that the typical American diet, which is a Western diet, is not good. It's much better to have a vegetarian diet. So that is an American study. They say it's not good for your health. But the fact of the matter is, if you look at then the comparison should be made between two very healthy individuals. Because if you look at it, you see, when I spent my years in USA, I found the Americans, Shahebs, are much healthier than we are. So if they are eating that diet and they are very healthy and they're winning all kinds of medals in Olympic, then it's a very difficult answer. But on the face of it, this paper said, and not this paper, number of papers said the vegetarian diet is better for your health. But
Each
then
food
again,
affected.
as I mentioned, this is one of the factors because you see that normally, even if you forget about the malnutrition, the average Indian, if you compare with an average, let's say, uh, Western country person, you can see the difference immediately. So there are other factors also involved, but from purely from the point of view of gut microbiota and all that, it's found that it is the beneficial microbes are more when people go for vegetarian diet.
Sir, excuse me,
Thank
one
you, sir.
question is giving on chat box. A student Rishabh Kaur, Department of Microbiology, put the question that, sir, you said that children having autism contain a particular microbe in a greater amount. Can we
Yes.
treat that individual by altering his gut microbiota even after his childhood gets over?
No, see, that's why I said that this is very controversial, because what happens, the companies which sell probiotics like Danone, then Yakult, they all jumped because they tried to hard sell it. They said that you have these probiotics and it will help autism. But the fact of the matter is, it is not so simple. This was one experiment which was done, a comparison was done with just 58 people, 58 children. Now the question is it true for for the children of different ethnicity, children from different places, children from different levels of nutrition? These answers were not known. So why it became controversial is just because it gave a handle to those commercial houses to and if you are the parent of an autistic child, you will try to cling to anything that gives you hope. So that is why it became so controversial. Probiotics definitely help, because even that today, even the doctors prescribe probiotics when they give you some antibiotic and all that. That part is known. But whether there is an one-to-one correspondence, like if I have typhoid and I take chloromycin, chloramphenicol, I get cured, that correlation is not there. So a lot more work needs to be done to actually see how far that is true. I mean, in general, it can cause some health improvement, but whether in a specific disease like autism, but giving those microbes will definitely help the children, this question is open. And the reason it became controversial is because of that. Because, see, the parents of autistic children, they are desperate people. They will like to do anything to give their children a normal life. So what happened? The probiotics sales went up and all of that. So therefore, there's no harm probably, but whether they'll definitely improve, the jury is still out.
Good evening,
Sir.
sir.
Good evening.
Sir, is there any vaccine to increase gut bacteria or to repair?
No. I mean, how can you have a vaccine to increase gut bacteria? You see, in vaccine, what happens? You try to generate antibodies, or you try to stimulate the T cells, and they do the job. So, if from that point of view, you can consider a probiotic to be a vaccine, because if you take probiotics, then you are flooding your system with good bacteria. So vaccines, what they do is they give you protection against invading pathogen by creating antibodies. And here what you are doing, you are replenishing the good bacteria. Our soldiers are facing the Chinese in the Ladakh border. So you send more soldiers to protect it's something similar to that. But there is no vaccine can be done in this.
Yes, sir. Thank you.
Hello, sir. I'm Shantan Vera from MSc Zoology.
Yes.
Sir, you already mentioned that three hypotheses for how microbes in the gut may talk to neuron in the brain.
Mm-hmm.
So my question is, sir, as basis on the second hypothesis, if we think and hypothetical model on who suffering with a wound in stomach, so that's why in this case many immune cells can release, and in this situation, if immune cell do action on those microbes already exist in our stomach, so then what will be the consequences?
No, let me try to understand your question once again. What you say is, can you repeat what you said?
So I told that if a model with suffering wound in stomach, and in this situation many immune cell can release
Suffering something in the stomach.
wound. Yes, sir,
Such
wound.
a wound, okay.
Any wound. So in this situation many immune cells can release, and that's why those immune cells do action on microbes already present in our gut or stomach. So if is so, then what will be the consequences, sir?
No, what do you mean by immune cells? See, you say
So
there's a
immune
wound in the stomach,
macrophage,
and
macrophage.
the stomach gets what happens is, as a result of that, the stomach tries to cure itself, and therefore, if it is macrophages and all this will come. If it's a bacterial infection, if it's a wound for other reason, then those have no role to play. So no, I'm really not getting your question correctly. So there is a wound in the stomach. As a result of that, body mounts immune response, and as a result of that, what happens?
Is there any chances that immune cells can destroy our gut microbiota?
No, gut microbiota themselves, induce the generation of immune cells. They generate cytokines, and those cytokines get into the bloodstream, and from the bloodstream they go into the brain, and then that is the model. Now here it is not 100% proven, but there is enough evidence to propose this. So therefore it's the gut microbes with the interaction with the cells which are lining the epithelium, the immune cells, because largest number of immune cells are present in the gut, they induce the generation of cytokines. So it's already there, and that is how it is going into the brain through the bloodstream.
Thank you. Another one question in chat box, that one of the cause of autism is genetic disorder. So does it means that genetic play a key role in gut microbiome formation?
Very good question. There is evidence that gut microbial composition, genetics do play a role. Now, depending upon what exactly it does, that is still an open question. People have just found that it has some component, genetic disposition or genetic makeup has some influence on the gut microbial composition. But exactly the one-to-one correspondence, to the best of my knowledge, has not yet been established. But it does have a role.
Okay, thank you very much, sir. So with
Yeah.
this, we come to the concluding.
Okay.
Okay.
Thank
Any
you
other
very much.
question?
And
Sir,
yes, please.
is there any evidence that same gut microbes show different moods in different groups of people?
You recall that bulb C mice experiment? So what happened was the bulb C mice is timid and NIH mouse is bold. So when they took out the gut microbiota from the NIH mouse and put into the gut of the bulb C mouse become bold. So therefore, if nobody has done any such experiment in case of humans, no, not correct. I will correct myself. See, there is a treatment method which is called fecal transfer. See, there are people who suffer from a disease called irritable bowel syndrome. This is a very intractable disease. People suffer diarrhea, all kinds of problem happens. So the only successful treatment is when the stool from a healthy individual who is not suffering from any disease is taken, made into a pill. You know, Indians will be very reluctant to do this, and given to a person whose bowel is first cleaned, and then this fecal matter is transferred to that person's bowel. Irritable bowel syndrome largely disappears. So, in answer to a question, that if you can replenish or replace the gut microbiota of a sick individual with that from a very healthy individual who suffer from no disease, depending upon the disease, you see great improvement. But that does not mean that if I have a brain disorder, this will produce the same effect. Maybe it does, but nobody knows it. But this fecal transfer experiment is now well established. Now, in Western countries, they make pills out of these stools, you know, which you or make it like coffee and people drink it. But we Indians are very reluctant to do it. So here it is done through operation. In all Indians of medical sciences, they do it. So they transfer through a surgical route, not like although the better method is if you drink it like coffee. But we Indians are reluctant to do that. So the answer to your question is yes, depending upon the disease, it can be done and it is happening.
Okay, thank you.
Most welcome. So let me just say that I have to send you two things. I have to send you the slides along with appropriate references, and also I have to send it that reference to the paper which talks about the evolutionary aspects of microbiota. This I'll send it to Swamiji, and then Swamiji you can take it. Yes, please.
Sir, you mentioned that there are pathogens or ectoparasites which can regulate our behavior. So how it is go on, sir? How can it regulate our behavioral mode?
No, so see, the question is, you're talking about the Toxoplasma gondii experiment? Is that what you're talking about?
Yes, sir.
The question is how it does it. Okay, now the point is how exactly does it is not known as yet. But only thing is known that even when the no trace of Toxoplasma gondii is in the system, still the behavior changes. So that means a permanent alteration has taken place, which means it must have produced some chemical or something which is able to do this job. Now, what exactly this chemical is still not known. But the method would be the same, probably would be the same as the three hypotheses that has been given. It must be doing something which is, I will assume that it must be producing either oxytocin or serotonin, which are the ones which has given the feeling of well-being. So therefore the fear disappears. So I will guess that, but what exactly the molecule is which is doing it is not known as yet.
So actually, this is the last. Actually, I tell the student has many questions, but sir has talked more than 1.5 hours. It is not a matter of joke. Sir, advances sir talking. Afterwards, when the situation will normal, I actually request sir to visit our college. Then student actually very much get benefit from it.
I will love to do that, Maharaj. I have never been to Roda Ramakrishna Mission. I can see the picture of the building. But I would love to do that. Let this COVID situation improve. So someday I will contact you and
Yes, sir.
come over.
Yes, sir. Yes, sir. Yes, sir. So I request my colleague Brahmachari Parachaitanya, please give the vote of thanks because after that, sir will take rest.
Yes, Maharaj, yes, Maharaj. So on behalf of our college, of behalf of our all teachers, students, and all entire college, I extend a really heartfelt gratitude to you, sir, for expending your huge time from your busiest schedule to grace the occasion and to deliver this wonderful talk. And I also thankful to you for this wonderful presentation and the interaction for our students. Today we had an opportunity to hear your thoughts, your ideas, and this will surely be going to encourage us, and your talk is surely be great inspiration to all of us present here, the students, faculty members, researcher who present over here and the YouTube channel. And we will hope that sir will definitely enlighten us again, and we really indeed to you, sir, for sharing your wisdom. And after this pandemic is over, we invite to you, sir, please come to visit our college and please enlighten us again with your wisdom and with your ideas. It was very wonderful talk. So on behalf of all, we extend our gratitude and thanks to sir for inspiring us. And thank you very much.
So
Thanks of all
all I
students,
would like to
teachers, all
say,
the
first of all,
participants. Thank you.
I don't deserve all that you have said. I have been a teacher all my life, so I love to teach and love to talk. And secondly, I felt deeply honored to be invited by an august institution like yours. I had an occasion to give a talk in Narendrapur, and Ramakrishna Mission Institute of Culture. But for me, it's always a matter of great honor when great institutions call me to give a talk. And you don't have to say all of that, all that you have said.
Yes,
I'm
sir.
just a person like all of you, born a little earlier, that's all. So in course of life, I learned something which all of you will learn one day. And I'm sure some of you will make contributions in the research. One thing I would like to say, because I'm a teacher, that please do read books which is not necessarily your textbooks, because see, when a scientist makes a discovery, it's a process, and you will know how this happens. This information we never get when we only read our textbooks or only go for our multiple choice questions. And it is extremely exciting. And some of the books, if you read, I can say they're unputdownable. The evolution, when Miescher isolated a white compound from pus samples, from there to here, DNA and CRISPR and all that. This is a journey which is fantastic, and that is something, if you read, then what will happen, you will feel excited about science. Because if you just do some experiments in the lab, run some gels and do it, you never get this excitement. In a jocular mood, I tell my students that, look, when you are sitting around the table in a canteen and try to talk, and you find that your adda is going nowhere, then somebody says, Ghosh sir to theek hai, he's okay, but he has this problem. Then you start discussing Ghosh sir and you find the time flies, which means what? Which means that we are all interested in human beings and human activities. And science is a hugely involved human activity, extremely exciting. But the way we teach, way we conclude give our textbooks and all that, this part is missing. And that's why many people will say science is dry. It is not. So my request to all of you, like, there's a book called Microbe Hunters. It's a very old fashioned book, but just read it. Rene Dubois, you'll see how these major discoveries were made. And then subsequently all of this is the only request I would like to make, and that makes science very interesting to you, not just by running some gels and learning from Watson's book how the transcription takes place. That misses out the human side of the story. So that's the only thing I'd like to tell you. As an
Okay,
old
sir.
man,
Thank you. Thank
as
you,
an
sir.
old man, I have this habit of giving advice. So I thought I could do it.
No, sir, it is very necessary. Otherwise, this give the proper direction. Otherwise, the student in future they could not take a decision just which path it is good for a good researcher or how
Actually,
to.
actually, Shamiji, I'll tell you something. You know, I'm sure that your institute must be different from that. You see, in most places, I see they neglect the libraries. I work in a national laboratory here, and students don't go to the library because library is not attractive. The books are not there. See, when I was a director of FinTech, you know, we had a fabulous library, and people will say, "Why you buy so many books?" I said, "Look, even 10% of our students read these books. It's worth the money spent." And but today I find that it's a circular argument. The authorities say that we don't buy books, the students go, and students say, "Sir, why should I go to the library? Nothing is there." See, you can load down a paper from your computer and read that paper, but the human excitement, how this came about, you can only have some idea about it if you read the book or the history. And that's why today I decided to give you this entire narrative in a historical manner. You see how Bowman never could imagine that his observations made 140 years ago will bring us to this place. Or nobody could imagine that, when doctors found that the irritable bowel syndrome and others causes depression, that eventually it will come to a stage where people will discover a link between the gut microbes and the brain. And there's all parallel paths, and suddenly there's converged. So these are the things only one comes to know if one reads a little bit about what happened in the past. See, as the Newton said, now we only see the standing on the shoulders of the people who came before us. So this is the only thing I tell the students of today, that please, and there are 24 hours in a day, so you can spend 1 hour doing that. No harm will come to you. Anyway, I said my bit, and so give it some thought.
Yes, sir. And afterwards, you come and visit
Yeah,
and talk with
that
our
was the
students. Actually, this talking also give them good inspiration.
I'll certainly do that, Maharaj. After this COVID situation is over, I'll get in touch
Yes,
with
sir.
you and
Yes,
I'll
sir.
come over. Yes,
Yes,
surely
sir.
I'll do
Yes, sir. Yes,
that.
sir.
Okay.
So, with your permission, we are actually close the meeting. If you permit.
Yes, Maharaj. I am also logging off. Thank you and good night.
Yes, sir. Sir, thank you, sir.
Thank you very much, sir.
Thank you.